Hey guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about fixation and fixatives. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So before understanding fixation and fixatives, we need to understand why do we require fixation technique or why do we require fixatives. So suppose you are working in a pathology laboratory or you are working in a DNA fingerprinting laboratory. So you have a tissue. You have a tissue from a diseased person or you have a tissue from a dead person. So you need to check something, that something fishy has undergone in the cell, in the tissue of that particular person's. So maybe the tissue is from a diseased person. So you need to check something that is there inside the cell. So what is happening? What is the fishy thing or what is the abnormalities that is occurring inside the cell? So for that you are isolating the tissue from the person's body. So now you need to check it under microscope. So the thing is, if you need to check a tissue or a cell under the microscope, you have to stain that tissue, right? You have to stain that cell because cells are transparent. So you cannot properly see that what is happening inside the cell under the microscope. So you have to stain that cell so that it makes a wonderful contrast, right? And second thing is, the cell is very delicate. Human cell is very delicate. So you need to make that cell or tissue a little bit hard. So for that you need to do one thing first. Before undergoing the staining process, you have to fix the cell. So what is fixation? You have to fix. So from the name itself, from the name fixation, you can understand that you need to fix something in the cell. And here you need to fix proteins. You need to fix proteins inside the cell. Now why you need to fix proteins inside the cell? Because the proteins are the culprits. Because when you are isolating or extracting a tissue from a human's body, the tissue will start decaying because it is now not in the human body. So it will start to decay. And this decaying process occurs because of the enzymes or proteins which are found in the cells. Those proteins and enzymes are the culprits which help in the decaying process. So you need to inhibit that decaying process. Because if you don't inhibit the decaying process, you cannot stain them, and if you stain them also, you cannot see anything. You cannot see the proper thing what is happening inside the cell. So because of that, you need to fix the proteins which are present in the cell. So that is fixation. That's why we require fixation. And fixatives are some chemicals. By using those chemicals, we can fix the tissue or cell. So now what is fixation? It is a technique by which the constituents of cells and tissues are fixed. So here constituents of cells and tissues, that means these are constituents, means these are proteins. Mainly you need to fix those proteins. Fix means you want to, whenever you are fixing the proteins, you are making those proteins inactive. You are making those proteins inactive so that they cannot do anything inside the cell. So it is a technique by which the constituents of cells and tissues are fixed or preserved in a physical or chemical state. That means both of its physical and chemical states are fixed inside the cell so that they can withstand subsequent treatments with various reagents without any decomposition or distortion of morphology. So what is the subsequent treatment? So here I have mentioned that subsequent treatment is the staining. But not only the staining, you can do some other things also, but you need to fix the cell first so that you will not expect any decomposition or distortion of morphology. Performed by treating the tissue with chemical compounds, those are called fixatives. So fixatives are some chemical compounds which we are using for this fixation purpose. Aims of fixation. So what are the aims of fixation? Why do we need to fix a cell or tissue? So to preserve the tissues to their living state. To preserve the tissues to their living state. Otherwise, the proteins will act and the proteins will start to decay the cell and the cell will not be anymore in its living state. So we need to preserve the tissue to their living state. To prevent autolysis and bacterial attack. So autolysis. So from the name itself you can understand this is the self destruction of cell, that is autolysis. This happens when you are removing the cell from the body. So cellular proteins or cellular enzymes will start to work and they will destroy the cell. That is autolysis. So we want to prevent that autolysis and we want to prevent bacterial attack also. To harden the tissues, we need to make the tissues hard so that they can withstand the further subsequent treatments. Prevent tissues from changing their original size and shape. We do not want the cell to change its size and shape, so that we need to add fixatives to allow good staining of tissue. And if the cell is in its proper state, if the cell is in its proper shape and size, it can take good stain and we can check the cell under the microscope in its proper form. To preserve tissue volume, we need to preserve the tissue volume. Otherwise, if the tissue volume decreases, then it is difficult to check the cell under the microscope. Now we will see what is the principle of fixation. What is the principle? So fixatives have a property of forming cross links between proteins. Hence keep them in their exact locations. So here I have told that we need to fix the proteins in their exact locations. So to keep them fixed, we will add fixatives. And what fixatives do? They cross-link different proteins. Suppose these all are proteins in a particular cell. Now I will add So now I will add fixative. So this is suppose the fixative I am adding here. So what it does, fixatives are some chemicals that will cross-link these proteins so that these proteins are bound, they are stuck in the particular location of the cell. So that is the principle of fixation. Next is the properties of fixatives and factors affecting fixation. So what are the properties of a good fixative and what factors they need? So first is that penetration rate depends on molecular weight of the fixative. Penetration rate of what? Penetration rate of the entry of fixatives in the cell. So if the fixative has high molecular weight, it will take more time to penetrate the cell. Similarly, if the fixative has low molecular weight, it will take less time to penetrate the cell. And the pH of the fixative should be between 6 to 8. The pH of the fixative should be between 6 to 8. Why? Because the physiological pH of the cell would be 7 to 7.5, right? 7 to 7.5 is the physiological pH of cell. So if you apply an fixative that has less pH or that has more pH, so it will hamper the physiological pH of cell and that in turn will destroy the cell. So that is lethal for the cell. Room temperature, we can do the fixation at room temperature, that means at 37 degrees Celsius. But 30 to 40 degrees Celsius is okay for that, because the cells which are there in our body, the physiological temperature is 37 degrees Celsius. So it is better to do the fixation in 37 degrees Celsius. And the fixative you are using should be cheap. It should be inexpensive. It should be non-toxic so that the fixation cannot do any harm in the cell. It should be non-inflammable. It should be non-inflammable so that it cannot induce any inflammation in the cell. So these are some properties of the fixatives and these are some factors which are affecting fixation. Next is types of fixatives. So different types of fixatives are there in the market. First one is the most popular one, that is formaldehyde. So that is most commonly used fixative. So formaldehyde is a gas. It is a gas, right? Now 40% formaldehyde in water makes formalin, that is widely used. So formaldehyde is a gas. So how will you apply formaldehyde? That's why you have to make it solution. So 40% solution of formaldehyde in water, it will make formalin, and formalin is widely used as a fixative. Mechanism of action. So how this formalin works as a fixative? It fixes the tissue by adding to the side chains of lysine. Cross-linking methylene bridges are formed where two formaldehyde binding sites are close together. So how does it fix the tissue? So it basically adds to the side chains of lysine. Suppose these are some proteins, and proteins has different amino acids. So among those different amino acids, one is lysine. Suppose this is lysine, and this fixative will be added particularly to that lysine. So I will draw that. So this is the fixative. Now, cross-linking methylene bridges are formed where two formaldehyde binding sites are close together. Now, different formaldehydes are bound with lysine amino acid of different proteins, and the cross-linking will occur between them due to the methylene bridges that are formed by these formalin. So in this way, they will cross-link different proteins and they will make them stuck. Advantages of using this formaldehyde. So it is a cheap technique. This is a cheap technique. We can easily use formaldehyde. It does not over harden the tissues. It does not over harden the tissues, because if the fixative makes the tissue very hard, it is not possible for that tissue to take the stain. And this formalin does its job very wisely. It penetrates the tissue rapidly. So it penetrates the tissue very fast. Disadvantages of using formaldehyde. It can lead to dermatitis of the hands. So it leads to skin irritation and irritation to the nostrils due to fumes. Due to the fumes of formalin, it sometimes causes hand irritation, skin irritation and irritation to the nostril. Next type of fixative is alcohol. Example, ethanol and methanol, these are used for the fixatives. So, mechanism we will see first. So, they replace water in the tissue. Therefore, they disrupt hydrophobic and hydrogen bonds and expose the internal hydrophobic groups of proteins and alter their tertiary structure and their solubility in water. So what does that mean? First of all, alcohols, what they do, they replace water in the tissue. They make the tissue dehydrated. Now what happens in a cell? Suppose this is a cell and this is a protein in this cell, right? So this protein it has some amino acids which are hydrophilic in nature, right? Some amino acids of the proteins will be always hydrophilic in nature. So these amino acids will face the cytosolic portion of the cell. So this is the cytosol. So these amino acids are hydrophilic, and as they are hydrophilic, they will always face the cytosolic portion of the cell, because cytosol contains water and these amino acids are loving water, so they will face that cytosolic part. And this protein will also have some hydrophobic amino acids. So let me draw that. So these are some hydrophobic amino acids. These hydrophobic amino acids will always stay interior portion of the proteins because they are hydrophobic. They have phobia of water. So they will hide themselves and they will be in the interior surface of the proteins. Right? In this way, the proteins are arranged in a cell. Now what happens here? You have applied alcohol and alcohol replaces water in the cell. So now you do not have any water in this cytosol. No water. So these hydrophobic amino acids are now exposed, right? They are now exposed because they do not have any such phobia now, because the cell has very few amount of water now. And these hydrophilic amino acids, which made some hydrophilic bonds with the water molecules in the cell, they will also alter their bonding pattern. So it will change the overall shape of the protein, because now there is the alteration of hydrophilic and hydrophobic bonding of the protein because of the dehydration. So this is the thing. So now the tertiary structure of the protein is destroyed, or we can say altered. As the tertiary or 3D structure of the protein is altered, the proteins will not function properly. And when the proteins will not function properly, there is no chance of decaying. Next, we will see the advantages of using alcohols. So it is the faster procedure. It is faster procedure. This is very fast procedure, and good preservation of cellular architecture. So by using this alcohol, we can preserve the cellular architecture. Cellular architecture means the cellular constituents. And the disadvantages of using alcohols: it removes water too fast from the tissue and leads to shrinkage of cells. Slowly penetrates the tissue and over harden the tissue. So alcohol removes water, but it removes water too fast, so that the cell is going to be shrink. And when the shrinkage of cell occurs, it is not easy to stain and you cannot check the cell properly under the microscope. And alcohol slowly penetrate the tissue. It takes time and it will over harden the tissue. So that is also not good for the fixation. Next is acetone. So acetone has the same mechanism, same advantages and same disadvantages like alcohols. Next is oxidizing agents. So oxidizing agents, we use osmium tetroxide. So what is the mechanism of it? It reacts with the side chains of proteins and allow them to form cross links that stabilize tissue structure. So these oxidizing agents, they will bind, they will react with the side chains of proteins, and then they will allow them to form the cross links, and then it will stabilize the tissue structure. So suppose here we have some proteins, we can see here we have some proteins, and now I am adding the fixative here, that is osmium tetroxide. So this is the fixative here, that is the osmium tetroxide, and this fixative is going to be attached with the amino acids of these proteins, and then it will help to form the cross links between these proteins. In this way it can preserve the cells and it can preserve the tissues. Now advantages of using oxidizing agents. So good preservation of cellular structures. It helps to preserve the cellular structure. It is used when samples are prepared for electron microscopy. So please try to understand that because this is important for your exam point of view also, that in electron microscope which fixative we use? We use this osmium tetroxide. The disadvantages are they cause extensive denaturation. So they cause extensive denaturation, and extensive denaturation is definitely not good for the cell. Next is picric acid. So the mechanism we will see here, they precipitate proteins by binding with the basic amino acids. So picric acid specifically binds to the basic amino acids of proteins, and then it can precipitate them. The advantages of using picric acid are they are good for connective tissue. If we need to check something in connective tissue, we can use picric acid. Disadvantages of using picric acid: so tissues cannot kept in picric acid more than 24 hours because it is little bit toxic. So that is the disadvantage. And one thing is there, you need to know that picric acid is used to make Bouin's fluid. Bouin's fluid, it contains picric acid, glacial acetic acid and 40% formaldehyde. So that is one more fixative. It is a compound fixative because it has more than one component. So it is containing picric acid, glacial acetic acid and 40% formaldehyde. The advantages of using this compound or Bouin's fluid is rapid and even penetration occurs without any shrinkage. So rapid and even penetration occurs without any shrinkage if we use Bouin's fluid. And the disadvantage of using Bouin's fluid is it is used for preservation of testicular biopsies. So this is used for preservation of testicular biopsies. So this is mainly used to preserve testicular biopsies, the samples which are going for the biopsies and the samples which are we are isolating from the testes. So mainly those samples are fixed by using these types of Bouin's fluid.